Hi, good morning, everyone. I'd like to welcome you to slightly more basic science-oriented rounds today. I'd like to welcome one of our previous residents who has quite an extensive background in research, even prior to becoming one of our residents. Stefan completed his BSc in Anatomy and Cell Biology at McGill, where he also went on to complete a MD PhD program and worked on the role of collapse and response mediator protein in mitosis and nerve regeneration as part of his PhD in neurological sciences. He completed his residency with us a few years ago and has now spent time at Harvard, Mass Eye and Ear, doing his work in cornea. And he's now joining us at Sunnybrook Hospital, where he's going to spend a significant part of his time engaged in basic science research on the cornea. So Stefan, welcome. It's a pleasure to have you at Sunnybrook, and it's a pleasure to hear about this fascinating work you're doing on the cornea. So thank you.
Thank you very much, everybody. So I'll give my talk in a little bit, but first we got one of our residents, Marco, that's going to be talking a little bit about some clinical, a little bit more of a clinical talk.
For those who don't know Marco, he's not just a resident, he's one of our star residents. He's a research machine.
Thanks so much, Dr. Rai, Dr. Lloyd, for the introduction. Thank you, Dr. Ongton, for your support in preparing this talk. Really excited to be able to discuss what's very much a novel kind of surgical technique in the armamentarium today for Fuchs endothelial dystrophy, namely descemetorrhexis without endothelial keratoplasty. So just to get going here. Slides. So these are just some financial disclosures that Dr. Ongton has provided for this talk. And so to begin with Fuchs, Fuchs is the most common condition of the corneal endothelium, characterized by the formation of these guttae or profibrotic collagenous deposits over the endothelium, with a significant predominance over the central endothelium as opposed to the peripheral. It results in visual disturbances, including blurry vision, halos, glare, and other visual phenomena. It's genetically heterogeneous, and there's a lot of work being done now to identify the genetic markers associated with this condition, which Dr. Ongton will discuss in a moment, the most common of which is the transcription factor 4 repeat expansion. With respect to Fuchs, typically there is a spectrum of disease, as you might imagine. First, medical therapy can be considered, whether that's hypertonic saline or steroids have been investigated, etc. But for more severe cases, surgery is typically needed. And currently, the standard of care is either DSEK surgery or DMEK surgery. And so your DSEK surgery is the Decimase stripping with automated endothelial keratoplasty, where it's a lamellar corneal transplant. The posterior part of the posterior stroma is removed along with the Decimase membrane and the endothelium. That's in contrast to a slightly thinner graft with the DMEK surgery, only corresponding to the Decimase membrane and endothelium being removed, and then a graft implanted. DSO or DWEK, these are two different acronyms to describe a similar concept. The first is Decimase stripping only, and the second is a descemetorrhexis without endothelial keratoplasty. So looking at these techniques without the use of a donor graft. So a central descemetorrhexis is made, and the theory behind this is that peripheral cells can migrate centrally to fill in the lost endothelium and ensure corneal clarity. And so just to go into how DWEK actually started from a few case reports, as most good surgical innovations do, from previous experience of the standard of care surgery. And so this was a case report here of an 80-year-old female with low visual potential at baseline, had a history of AMD, who had significant Fuchs and underwent DMEK for that. And unfortunately, just hours after the procedure, had a free floating roll in the anterior chamber, predictably developed corneal edema for a few weeks, which improved over the course of weeks to months, and had eventual normalization of both vision and tachymetry at 6 months. So this was really one of the foundational cases to suggest whether donor corneal endothelial cells on the carrier Descemet's membrane are sufficient to repopulate the endothelium of the recipient posterior stroma. And another novel case that came out in 2012 as well was that of a younger female, 34 years old, with a mixed presentation of a polymorphous versus a Fuchs-like presentation. The history here was that this lady underwent DSEK in one eye, which failed, and then underwent revision surgery. That revision surgery postoperatively was seen at the periphery to be starting to detach. And with each subsequent visit, what happened was the detachment continued to extend slightly from the periphery to the center. And what the authors noted was that there was actually in previous areas where there was peripheral edema and peripheral detachment, those areas eventually started to clear, again in a pattern from the periphery to the center of the cornea. And so this at this time was a novel observation, and the hypothesis was, well, perhaps some of the peripheral corneal endothelial cells are compensating for the loss in the center, and perhaps there's actually migration of these peripheral cells more centrally. And so, you know, when you looked at a more long-term time frame, by 6 months, this patient had done actually quite well with an excellent visual outcome and corneal clearing. The final case was an unfortunate one of a routine phaco being done in an 84-year-old woman who had unfortunate viscodissection of the Descemet's membrane with a 4.5 millimeter decimeterexis performed. Again, the initial significant corneal edema, which eventually cleared over months and had an overall final visual outcome. And so now we were seeing kind of different cases from different centers postulating whether a simple decimeterexis alone could be one of the options for Fuchs. And that's really where you know, DWEK surgery was born. And so now there's a little bit more known about the procedure and its indications. Specifically, it's for now indicated for Fuchs with central guttae. Typically, the 4 millimeter central zone needs to be involved, and they need to have enough of a peripheral endothelial reserve. When you look at the literature, typically authors quote at least 1,000 cells per millimeter squared on the microscopy for this procedure to be considered. Obviously, they have to be symptomatic enough. They have to otherwise be a surgical candidate, and the periphery has to be cleared, as I mentioned. The lens status really does not have any bearing on whether this surgery can be performed. So just going into a case presentation that Dr. Angton shared with me, that of a 63-year-old gentleman who had Fuchs, was symptomatic with morning blurring and photophobia, was pinholing otherwise at 2025, myopic with some astigmatism, had normal pressures and 660 central corneal thickness. He had central and paracentral guttae, but his periphery looked quite good. And when you looked under the confocal microscopy, you could see centrally he almost had no endothelial cells, whereas peripherally he actually had quite a good reserve, making him a good candidate for this procedure. So just going into a surgical video here of how the DWEK actually looks, calipers are used to make a 4 millimeter marking here before the procedure begins. And you'll see in a moment that reverse Sinskey hook is used to make a 2 clock hour score to retract a small Descemet membrane flap, which will then be extended 360 degrees with MST forceps to create a decimeterexis. So we see the score being made here, and then the forceps used to extend the full rexis here. It's important when doing this procedure not to engage the posterior stroma, because that can lead to stromal tags and visual decline as a result of that. And it's also important to note that these are quite friable and weak folds here. And so if it does break, one just needs to engage the leading edge of the rexis that's performed and reflect back the remaining Descemet's to complete the rexis. This is all done under Huon OVP, which is removed at the end of the case. So looking at this gentleman's post-operative time course here, you see again the initial corneal edema with poor vision, eventually normalizing by about a month or two after the procedure with excellent final visual outcome and normalization of the central corneal thickness here. When you then look at his specular microscopy at about 1 month post-operatively, he did demonstrate a more healthy central endothelial reserve, again suggesting this notion that peripheral endothelial cells may have migrated more centrally. And so then the question becomes, well, should DWEK be indicated for all patients? The experience thus far has been that this is sometimes an unpredictable procedure, with some patients failing to achieve perfect corneal clearance. And so I just wanted to include a couple of factors that may mediate clearance in this setting. The first is the dysmetorexis size. So for corneal surgeons that do DMEK, the typical graft there is 8 millimeters or 9 millimeters, so quite a larger dysmetorexis size rather. With the DWEK surgery, a smaller dysmetorexis should be considered of slightly less than 4 millimeters. And so research has shown with these larger grafts with like an 8.5 millimeter dysmetorexis, there are about 40% of the corneal endothelial cells that are lost, making it more difficult for peripheral endothelial cells to migrate in centrally, whereas with a 4 millimeter dysmetorexis, only 10% of the total endothelial cells are lost, making it more likely for better peripheral migration. The surgical technique also needs to be considered here. So when you're doing that scoring, as we saw in the surgical video, only 2 clock hours were done with the reverse Sinskey to score initially, and then the forceps were used to create that 360-degree dysmetorexis. That's in contrast with the typical DMEK surgery, where 360 degrees of scoring would actually be performed. And researchers have looked at this, having that only 2 clock hour score is actually associated with better corneal clearance. And with the 360 approach, there have been reports of stromal scarring, small decimated membrane detachment, and edema that have impeded the success of this procedure. Patient factors are also important to consider. Unfortunately, pachymetry is only weakly correlated to final outcome, though on microscopy, the central endothelial cell count and peripheral endothelial reserve is important to document and to be at least over 1,000. And then ex vivo observations have also shown that patients who are younger, who have an intact decimated membrane at baseline, those are patients that do well. Recent research has looked at the supplementation of Rho kinase inhibitors. And you may remember Rho kinase inhibitors from the glaucoma sphere, where they have been shown to lower episcleral venous pressure and reduce the amount of fibrosis on the TM, leading to improved outflow. Here, their mechanism is a little bit different. So I realize this is a complicated slide, but the main thing to note is that the ROCK inhibitors currently have been shown to inhibit this P27-KIP1 factor, which is essential for the inhibiting the normal cell cycle. And so as a result of that, there's increased proliferation of endothelial cells, perhaps even increased migration of peripheral cells centrally, and an inhibition of apoptosis. This has traditionally been used, ROCK inhibitors have been traditionally used as a salvage agent, though there is an ongoing placebo-controlled randomized control trial now, which is looking at the role of DWEK plus ROCK inhibitors at baseline versus DWEK alone to see whether the rates of corneal clearing are any different. Postoperative care otherwise is, as you would imagine, with a topical antibiotic and steroid. Hypertonic saline can be considered in cases of significant corneal edema, and the Rho kinase inhibitor, as we mentioned. So just summing up, DWEK is still an early technology, but it may have a role to play in the surgical armamentarium for Fuchs. Its advantages is that you do not require a donor graft, and so you don't need long-term immunosuppression. There's not a chance of graft rejection, failure, detachment, etc. And if one wants to later on, they can perform an endothelial keratoplasty if the original procedure fails. And right now it's a little bit uncertain as to when specifically that should be done. Anecdotally, based on what Dr. Ong Tone has seen, it's been anywhere from 3 to 6 months after the procedure. If the cornea really hasn't cleared, then an EK can be considered. There are limitations of the technique. Case selection needs to be made in a very thorough fashion. There are complications associated with creating the descemetorrhexis, having it be decentered, irregular astigmatism, non-resolving corneal edema, some of those stromal tags if you engage the posterior stroma. And right now, the data is unfortunately for this procedure limited to case reports and small case series. I just want to thank you for your attention, and I'm looking forward to hearing Dr. Ong Tone's presentation on this subject as well. Thank you.
All right, thank you, Marco, for that great introduction. I'm just going to share my slides here. All right, can you all see that? Assuming that's a yes. All right, so today I'll be talking a little bit about my research that I performed over at Mass Eye over the last 2 years, and this was in the lab of Dr. Uller Kunis. So before we start, I'm just going to show you this movie over here. It's playing in the background. These are green labeled, GFP labeled corneal endothelial cells from a Fuchs patients that are migrating on their native Descemet's membrane. So a question I want you guys to think about is, do you think that these Fuchs cells are faster or slower than normal cells? And hopefully through this presentation that you will have an answer. So these are the financial disclosures for the lab that I was in. So as Marco alluded to, that typically for surgical management of Fuchs endothelial corneal dystrophy, we've been treating it with through DSEK or DWEK, but more recently a novel surgical technique has been introduced called Descemet stripping only, also referred to as Descemet stripping without endothelial keratoplasty as DWEK. So this really relies on central descemetorrhexis and the migration of these quote unquote normal peripheral endothelial cells that will migrate and repopulate the central endothelium. So a little bit about corneal endothelial cells in general. So they're this monolayer of hexagonal cells that are derived from neural crest cells, and they rest on a specialized basement membrane called Descemet's membrane. They're post-mitotic cells arrested in G1 phase of the cell cycle, and they typically do not proliferate in vivo. We know at birth that the endothelial cell density is approximately 4,000 cells per millimeter squared. These studies were done here at UofT. We know that in an adult that there's a steady decline in just normal healthy endothelium of approximately about 0.6% per year. So what is the wound healing response in an adult human corneal endothelium? So we know that there's a limited role for cell division and wound repair in corneal endothelial cells. And the wound repair mechanisms really are kind of occur in a stepwise fashion. In small sort of wounds, and you can kind of think of cell death leaving kind of an area devoid of corneal endothelial cells as a small wound. And typically these cells will repair the wound by enlarging. And the adjacent cells will enlarge their surface area and close that wound. As the wounds get slightly larger, we know there's a phenomenon called monolayer spreading. And that basically means that there's coordinated movement and closure of the wounds by adjacent cells by maintaining their cell-to-cell contact and migrating collectively to close that wound. And as wound ultimately gets larger, we actually see individual cellular migration to help close these wounds. And I think this concept of cell enlargement is something that I really, by kind of understanding more about this process, I think clinically what we see on specular microscopy of the corneal endothelium with this polymegathism and pleomorphism, this variability in cell size and shape, kind of recapitulates and shows this phenomenon, because we can see that there's different sizes of corneal endothelial cells, some larger, some smaller. And I suspect that these larger cells are cells that are undergoing this cell enlargement process. So what do we know about Fuchs and corneal endothelial cell migration? First, if we focus on normal corneal endothelial cell migration, what do we know? So we know that it's dependent on extracellular and intracellular signaling, that it's also a process that's dependent on cytoskeleton rearrangement, specifically the actin and microtubule cytoskeleton. We know that Rho GTPases are important regulators, and we know one of the downstream effector proteins, ROCK, inhibition actually promotes migration. What do we know about Fuchs, specifically Fuchs and corneal endothelial cell migration? Well, there's actually not much data out there on specifically Fuchs cells and corneal endothelial cell migration. And much of the findings that from normal corneal endothelial cells are actually extrapolated to Fuchs corneal endothelial cells. And part of the reason that there's lack of data on Fuchs corneal endothelial cell migration is that it's actually very difficult to grow and to study these corneal endothelial cells derived from Fuchs patients in the lab. And additionally, we know that there's limited access to patient specimens undergoing endothelial keratoplasty. So, and there's actually limited models to study Fuchs. So the purpose of my study was to really study corneal endothelial cell migration in Fuchs specimens and Fuchs patients themselves. So the methodology that I've developed is a novel live imaging cell migration system where we look at Fuchs and versus normal cadaver corneas and corneal endothelial cells. And I've generated these immortalized cell lines derived from either Fuchs patients or normal donors. And we look at their migration patterns under low cell density conditions and high cell density conditions. Additionally, we can look at ex vivo specimens. So looking at the specimens, these are specimens that have derived from patients undergoing DWEK. So I would take after surgery, I would take these specimens directly to the lab where we can perform these experiments. And we look at areas on the specimens of low cell density and high cell density and looking at differences in cellular migration. And we've also, I'm also developing a DWEK DSO model to study cellular migration. And to study cellular migration, we use this automated analysis of cellular migration using this TrackMate plugin. So we've created these Fuchs cell lines that have been immortalized. So again, what we do is, so actually patients are undergoing DWEK, we take their tissue and we actually take them to the lab and push them in tissue culture. And we can isolate the corneal endothelial cells from these Fuchs patients and can immortalize them using this SV40 virus. And what this really allows us to do is have a very valuable tool of these Fuchs-derived corneal endothelial cells that we can now use for many different experiments. Because one of the challenges of studying Fuchs is that if we just take the corneal endothelial cells and try to grow those in culture, we can only passage them for a few times and only do a few limited number of experiments before the cells die off and senesce. So this is, these are very valuable tools. And we can actually generate many different cells from different patients where we have normal patients and different Fuchs patients. And also at the time where we collect the specimen, we also consent the patients and collect their blood. And this allows us to do genetic analysis of, from these blood samples. So we can actually identify you know, what mutation that these Fuchs patients have to the corresponding tissue. So for these experiments, I'm going to be showing you data from one normal cell line and two Fuchs cell line, one of which has this TCF4 mutation in the cell line that's, that was described previously by Marco. So this is actually the most common mutation in Fuchs, which has been reported in up to about 75% of patients. So what you're looking at here, these are live cell images, videos of normal and two Fuchs cell lines. And these are plated under low cell density conditions. And it's a time lapse over about 16 hours or so. And what you can see is that these, the two Fuchs cell lines here display increased cellular migration speed compared to the normal. And this is just a representative image here or video of how we do this automated tracking is that we, we identify the cells kind of on the beginning of the video, and then the program just tracks the movement over time. And from there, we can calculate migration speeds. So these are just micrographs here showing some of the parameters that we look at, looking at mean speed and maximum speed, and here's the actual quantification. And the quantification here showing is that these two Fuchs cell lines actually display statistically significant increased migration speed compared to the normal cell line. And similarly, if we look at mean maximum speed, these two Fuchs cell lines display an increased speed compared to the normal cell line. So these are under low cell density conditions. So what about under high cell density monolayer conditions? So to analyze these migratory properties, I performed a scratch assay to analyze cellular migration under these high cell density conditions. So the setup for this experiment is that these cell lines are plated in high cell density or monolayer, allowed to grow to 100% confluency over 24 hours. I perform a kind of basically a wound with a pipette tip. And then we watch how this wound closes over time through live imaging. So what you're seeing here on the top, so these are about 16 to 18 hour videos of this scratch assay. So you can see that with the normal cell line, at the end of the video, 18 hours, there's some closure, but there's not complete closure of the scratch wound. Whereas the two Fuchs lines, at the end of the video, you see that there's almost complete closure of the scratch wounds. So this data that I'm seeing displaying increased cellular migration speed under these high cell density conditions is consistent with what the previous observations that there's increased cellular migration speed under low cell density condition in Fuchs. So this is great. These are all in cell lines, but what actually occurs in ex vivo specimens? So to answer this question, we've used this live imaging system here, where we can fluorescently label all the corneal endothelial cells using these lipid nanoparticles to introduce green fluorescent protein to the corneal endothelial cells. And this allows us to better visualize the corneal endothelial cells and allows us to track actually through live imaging a little bit better. So there's a setup here is, so we take Descemet's membrane with the corneal endothelium. These are from normal cadaveric donors, and we put them in tissue culture. At the same time, or sequentially, we can take Fuchs specimens. So these are patients undergoing DMEK, and I can take these tissues directly from the patient after they go surgery, bring them to the lab, put them in tissue culture. So here they have the Descemet's membrane with the guttae and the corneal endothelial cells. And we can label these corneal endothelial cells, as I mentioned, with this green fluorescent protein. And this allows all these cells to be labeled with this GFP signal. So this is an example here of an ex vivo specimen. So this is about 8 millimeters that has been stripped. And you're looking at here after labeling, all these green dots here are individual corneal endothelial cells that have been labeled and now are expressing green fluorescent protein. So this is an example here of the live imaging setup. Basically, there's a membrane here that had been labeled here. There's an area here of corneal endothelials that have been labeled, and I can select different areas on this screen basically to image over time. And this allows us to image multiple different areas on one specimen to get the data. So this is what we're looking at here is these are live ex vivo specimens. So these have not been fixed, and you have low and high magnification here panels. So under normal, so this is a normal tissue. You can see that there's a lot of corneal endothelial cells that have been labeled. And if you look at under high magnification, you can actually see individual corneal endothelial cells here that have been labeled. In contrast here, when you look at the Fuchs specimens, obviously there's a lot less corneal endothelial cells because there's, this specimen or this patient has undergone a DMEK. So it's usually more advanced Fuchs, so there's more death of corneal endothelial cells, less density. And under high magnification, actually what's really cool here is that, so all these dots here that you're seeing under phase contrast, these are individual guttae or guttata. And these are the labeled cells. So you can actually see here this interaction between corneal endothelial cells and the guttata. And again, these are ex vivo specimens. So these are cells on their native Descemet's membrane. And if we fix the tissues, just to get an idea of, how efficient is the labeling. So this is normal and these are Fuchs. We can see here when we counterstain with a nuclear stain DAPI, which labels all cells, we can see that there's high transfection efficiency and good GFP expression in the majority of cells. Similarly, we see that although there's less cells here in the Fuchs specimens, that we get good transfection efficiency and expression of GFP in these ex vivo specimens. So just to go over the experimental protocol for the live imaging. So we take the Fuchs ex vivo specimens and these normal ex vivo specimens, and I try to select areas of high cell density and low cell density for each and perform live imaging. And before I show you the movies, just want to show you this really cool image that I took. So this is a phase contrast image of a Fuchs tissue. This and all these bumps here are actually not corneal endothelial cells. They're actually the guttae. And it's just a good reminder to show that there's centrally here, there's a high density of guttae, whereas as you go towards more the periphery, that you can see that the density of guttae kind of becomes lower. So I'll just show you a representative image here, a movie. So these are Fuchs endothelial cells labeled with green fluorescent protein, and they're migrating on their native Decimus membrane and interacting with guttae. And this was imaged over 16 hours. And I remember this was actually one of the first movies that I had taken. And I was actually so impressed with how quickly these endothelial cells migrate. And actually a little bit surprised because we're so used to looking at specular microscopes and thinking about endothelial cells in terms of morphology as this hexagonal compact layer. And really these cells didn't really look anything like it. And they actually, you can see that they actually become quite elongated and fibroblastic in nature over the movie. So what about if we look at these high cell density areas? So this is normal versus Fuchs tissue. And really there was no difference in cellular migration speed when we look under high cell density conditions. And this is probably because the cells are maintaining their contact to contact inhibition, and there's really no difference that we observed. In contrast, when we looked at these low cell density conditions in these ex vivo specimens, so this is in normal tissue, you can see that they're still migrating. But when we look at these Fuchs specimens, these areas of lower cell density, we can see that the cells are really migratory and really moving around a lot. If you, for example, like this cell over here moving around. So this is about over a 16-hour time period. So this is the quantification of the cellular migration, and there are really three take-home points that I want to highlight here. So the first take-home point is that either if you're looking at normal or Fuchs tissues, that if you look at high cell density versus low cell density areas, that there's increased cellular migration in the low cell density conditions compared to high cell density conditions. This is either in normal or Fuchs. That's the first point. The second point is that if you look at high cell density areas in normal and Fuchs, there's actually no statistically significant difference between the two. And as I was alluding to before, this is likely due to maintained contact, cell-to-cell contact inhibition. However, when you look at these low cell density conditions, that while both of them display increased cellular migration speed, that Fuchs compared to normal display a statistically significant increase in cellular migration speed. So this is all great, but you have to remember that these are cells that are migrating on their native Decimus membrane. And for Decimus stripping only, or DWEK, the cells actually migrate off of their native Decimus membrane and actually onto bare posterior stroma. So I wanted to develop a model to study this. So the model that we came up with was basically we take a DSEK button and we expose it to bare stroma. So this is what you're seeing here. And then we can take a stripped Decimus membrane from a Fuchs patient and actually overlay it on top of bare stroma. And we can do the same thing that we do for the live imaging. We can label all the cells with green fluorescent protein to allow us to better visualize these corneal endothelial cells. We're going to try to see here if we can get if we can visualize these corneal endothelial cells migrating onto bare posterior stroma, which would be similar to what occurs during DSO or DWEK. And so this is still a technique and model in development, but I just wanted to show you this video here showing that we're able to visualize some of these corneal endothelial cells actually here migrating off of their native Descemet's membrane and onto the bare stroma over here. So here you're seeing the edge of the tissue, and then here's the stroma, and you can see that over time that there are some cells here that are migrating onto this bare stroma. So unfortunately, we're still developing this model, and I don't have any good data to show, but this is a work in progress. So the question is, like WTF, like why the Fuchs? Why do these Fuchs cells display faster cellular migration speed? So I'm showing this schematic diagram to highlight here that we know that Fuchs is very complex, and we know that there's a lot of genetic, epigenetic, and exogenous factors that contribute to Fuchs pathogenesis. And a lot of work has been done in trying to understand Fuchs pathogenesis. And we know that oxidative stress plays a big role, mitochondrial dysfunction plays a big role. But one of the things that we know plays a role is this endothelial to mesenchymal transition. So what exactly is endothelial to mesenchymal transition, or also known as EMT? So we know it's a process that actually occurs in many different cellular processes, not just Fuchs pathogenesis. We know that it's important for embryogenesis. We know that it's important for cancer metastasis. So it's been linked to be very important for cellular migration. And there's this spectrum model here that's been proposed to look at the link between EMT and cellular migration, that as cells are more on the spectrum of this epithelial-like or endothelial-like characteristic, we know that they tend to have more of a collective migration pattern. As cells transition and basically lose some of these cell-to-cell contacts and become more individual cells and more mesenchymal-like, that we know that the pattern of migration tends to become more of an individual cellular migration. So what about EMT and Fuchs? So if we look at our cell lines and also specimens, and we look at EMT markers. So here I'm showing a Western blot of normal cells and the two cell lines here. And I've probed it for different markers of EMT. So edcoherent, fibronectin, SNAIL1, and smooth muscle actin. So these are all markers of EMT. And the cell lines display increased expression of these EMT markers. Similarly, if we look into specimens, so these are normal specimens here on this left side here, and these are Fuchs specimens. You're showing here by immunohistochemistry that their EMT markers here are increased by increased fluorescent intensity. And down here, if we look at by Western blot by specimens, we see that there's increased expression of some of these EMT-like markers, suggesting that overall that there's an increased expression of EMT in Fuchs. So the question now becomes, why do Fuchs cells undergo this EMT type process? So I think, if you look at these ex vivo specimens, so this is a normal specimen here that has been labeled with, to show the filamentous actin. And you can see that there's this tightly compact monolayer of these corneal endothelial cells with this beautiful subcellular actin network here. But in contrast, when you look at these Fuchs ex vivo specimens, you lose this monolayer and compactness of these corneal endothelial cells. And you see a lot of these corneal endothelial cells are now larger. And actually, when you look at the actin cytoskeleton, you can see that some of them are actually extending lamellipodia. And some of them display this more elongated now fibroblastic-like morphology, suggesting that these cells are undergoing EMT. Similarly, if you look at the Fuchs cell lines that I've generated, that they do display this fibroblastic morphology. So here I stained normal and these two Fuchs cell lines with ZO1 to show that they do form these cell-to-cell junctions. However, if you look at the actin network, at least qualitatively here, you notice that these two tend to display a more fibroblastic-like characteristic. So another piece of evidence from another experiment that I performed is I've looked at Fuchs versus normal whole transcriptome analysis, and basically performed RNA sequencing to look at all the differentially expressed genes in Fuchs. So to do this experiment, this RNA sequence experiment, I've taken 7 Fuchs cell lines that we've generated and compared them to 3 normal cell lines and performed RNA sequencing and subsequent bioinformatics. And this has identified 215 differentially expressed genes. And when you look at all these different genes and try to do this pathway analysis to see what pathways most of the genes can be assigned to, interestingly, the EMT pathway and cancer metastasis pathway come up as positive hits. So this is still a work in progress, and we're still going through some of this data, but it's interesting and exciting that these 2 pathways have been identified kind of in a non-biased way. So currently the working model for Fuchs pathogenesis EMT is that you have this pre-symptomatic Fuchs tissue. So these are patients that have this nice compact monolayer corneal endothelial cells and probably have some sort of genetic mutation that predisposes them to undergo corneal endothelial cell apoptosis. And as more and more of these corneal endothelial cells start to die off, it initiates this wound repair mechanism. And when you have early cell death and maybe just small wounds, the cells can compensate by cell spreading. So just adjacent cells spreading. And I think that's what we're seeing when we look at the specular microscopy and looking at and seeing the polymorphism and polymagnetism. Ultimately, I think what happens is that more and more of these corneal endothelial cells die off, and we can no longer cover the exposed decimus membrane just with cellular spreading, and we need to initiate cellular migration, first by monolayer spreading, but eventually to individual cell migration. And ultimately, this leads to symptomatic Fuchs with corneal endothelial cell death and corneal endothelial decompensation of the corneal endothelium. And I hope today that I've shown that EMT is a process that's involved with this, with Fuchs pathogenesis and cellular migration. And I guess one question is that, is this really a cause, or is EMT part of the pathogenesis, or is it merely a consequence of the corneal endothelial cell death that we're seeing part of Fuchs? So I'm showing this movie again here that I was showing you from my introductory slide. And I hope that I've demonstrated and supported the evidence that Fuchs corneal endothelial cells display increased corneal endothelial cell migration compared to normal. And the question that arises, what is the implication for clinically now for DWEK or DSO? And that remains to be completely elucidated. So in summary, I've developed this novel live imaging model to study corneal endothelial cell migration in Fuchs specimens. I've shown that Fuchs cell lines display increased cellular migration, that Fuchs endothelial cells on ex vivo specimens also have this increased cellular migration speed, particularly in low cell density conditions, that the cells display a pro-migratory phenotype, and that this may represent EMT. And I'm interested in understanding how this EMT is linked and its role in Fuchs pathogenesis. And I think it can have potential implications in corneal endothelial cell migration in DWEK or DSO. So in terms of future directions of my own research and where I'm going to be taking the direction kind of of my lab, is that kind of focused on trying to elucidate, the migration, what controls the migration speed in Fuchs. And there's a lot of different things we can look at. How does the central versus peripheral location of the corneal endothelial cells affect migration? How do Goutte density actually affect migration? So we know that, centrally there's a higher density of corneal Goutte. So how does that affect the migration? I'm also interested in further elucidating how this genetic mutation of TCF4 affects cellular migration. And as we were mentioning before, the ROCK inhibitors now are being used with DSO, DWEK. So how do ROCK inhibitors affect migration speeds? And I'm particularly interested in trying to understand this mechanistic link between EMT and Fuchs. So I've showed some data showing that with this RNA sequencing data, that potentially we could identify new candidate pathways and targets that have not been linked to Fuchs before. And I think what's really interesting clinically is this potential of, can we predict which patients will respond to DWEK or DSO? because even now with patients undergoing DSO, even with ideal patient selections, there are still some patients that fail to respond to DSO, DWEK the way they we anticipated to. So what's underlying, those lack of response to surgical therapy? And could this be the beginning of personalized medicine in cornea? And maybe we can genotype to predict phenotype, and maybe the TCF4 mutation or another genotype may be a predictor of cellular response and surgical response to DWEK, DSO. And one of the things that I'm going to be setting up here in Toronto is this biorepository of blood, aqueous humor, and specimens from Fuchs patients undergoing endothelial keratoplasty. And I think this would be a very valuable tool for us to perform some of these studies. And just thinking ahead kind of long term, I think one of the really interesting potential treatments that could be coming in the future is maybe new surgical treatments for Fuchs. So maybe the future is that we can isolate corneal endothelial cells from asymptomatic Fuchs patients. We can identify their genetic mutation, maybe genetically excise this mutation through CRISPR-Cas9. We can expand these cells ex vivo and maybe replace their abnormal cells with these genetically engineered cells. So this is, really exciting, and I would think that maybe it's really far down the line, but now with just recently Luxturna being approved by Health Canada for gene therapy for RP, so this is actually maybe more in our future than we anticipate. So with that, I just want to thank my lab and Dr. Ula Yerkunis, who is the PI and cornea specialist that I worked with in Boston, and different lab members that have helped me with different aspects of my research. And I'll put up the QR code here for your feedback for you to scan. And while that's up, I just wanted to show you this picture of me and my family. So that's my wife and my son. And they've been very supportive through my career as I've migrated from Montreal to Toronto to Boston. And now I'm migrated back here to Toronto to continue my research. And I'm very excited to continue this work here at the University of Toronto and at Sunnybrook.
So thank you very much, Stefan. That's some really fascinating work. There was at least one question from the audience. I'll just lead off with a question of my own. So was unaware that you could do a DSO and have the cells migrate in like that with good outcomes. That's quite amazing. Presumably though, since these patients have a disease, the disease will eventually recur in the area where the cells have migrated. Do you have an educated guess as to how long that may take to happen?
Well, I mean, if you think about how long it took for them to express the phenotype, usually you see them in their 60s. So I don't have a good answer, but I suspect that it should be a while. But I think it's just such a novel technique that we don't really know. I think you can look at least from maybe some of the data from the Kinoshita group in Japan, where they're actually doing cell therapy. So they're basically taking normal cells, expanding them ex vivo. So these are from cadaver donors, expanding them ex vivo, and they're removing the abnormal corneal endothelial cells from a patient, but they're leaving the Decimus membrane and guttae intact, and they're injecting these cells into the cornea, into the aqueous humor, and allows them to repopulate and reform the corneal endothelium. They, and one of the criticisms, has been, well, you're not removing the guttae, or you're not removing the extracellular environment. So will these cells sicken over time? And they've shown at least 5-year follow-up data with good results. So I would anticipate that the results from DSO would be good for if the patient's in their 60s, 70s undergoing the surgery, maybe for their lifetime, hopefully. But we don't know.
John, we've got patients who are now about 2 years out. We have not seen any gutata reform. And in fact, we're starting a multi-center trial, Canadian trial, which Stefan's going to be a part of. And Stefan, I wanted to ask you, can we do any studies in vivo using specular microscopy to look at the migration of these cells, both with and without, Glanatech? Is that a possibility?
So the challenge is that there's two basically challenges. Is how do you? I think one is an imaging challenge in terms of the technology. We do have obviously the ability to image at the individual cellular level, but the challenge is that getting the temporal sequence to get some of these images, because the videos that I'm showing are over 18 hours or so where we can see this migration. And I suspect that part of it is an artifact of examining these cells ex vivo, that with culture medium, they probably speed up a little bit more than what actually happens in vivo. But I think the challenge of visualizing individual cellular migration in vivo is that, how do we get a patient to sit there for 18 hours or, longer than that potentially, and then the alternative is that if we bring them back, for multi-day imaging, how do we identify the exact same cell? So it's something that's interesting, but I think technologically there's still some challenges there.
Yeah. The nice thing about doing DSEK or DSO rather is that if it doesn't work, and there's overall about an 80% success rate, is that you can always do a DMEK afterwards, whereas you can't do a DSO and then do or do a DMEK and then do a DSO afterwards. So you can always, and we've actually done that. We had a patient who didn't clear, and we did a DMEK, and that worked out quite nicely for that patient. Yeah, and just the only thing is that you got to tell patients in advance it's going to get worse, and it's going to stay worse for a month, couple of months, and then it's slowly going to get better. So if you, it requires a really in-depth sit-down conversation with the patient before going ahead.
Right, I agree. I think that's one thing that in terms of patient selection, the patient has to be in the right, obviously has to be informed in the right mindset to understand that it may take a little bit longer for the cornea to clear, but the advantage may be that they may not need a transplant and may not need lifelong immunosuppression with topical steroids.
Clinically, I found that the uptake has been very good with these patients if you talk to them in advance and they know what to expect.
Do these cells that migrate into the gap that you created with your DSO, do they reform any sort of basement membrane, Descemet's membrane, and then could you actually peel them off again in the future?
I don't know I would suspect they would, but I don't think we've actually looked at it histologically. Because these patients, they're still out there, and I think that would be interesting to look at, patients that had undergone DSO and then looking at their corneas and seeing if they reformed their Descemet's membrane. But I would suspect that there would be some sort of deposition of basement membrane. I don't know if it'd be the full Descemet's membrane, but I think that's an interesting question.
So some questions from the audience, from Dr. Lichter. Are guttae the result of apoptosis? Or I guess more generally, what are the guttae?
Yeah, so that's a $1 million question, I think, in cornea. I think there's always been this debate about what actually leads to guttae formation. Is it kind of like the chicken or the egg? So I think so the question is, whether these corneal endothelial cells secrete this guttae, this abnormal extracellular matrix that now subsequently cause corneal endothelial cell apoptosis, or are these really? Is there something intrinsic about these corneal endothelial cells that they're starting to apoptose, and as a result, they deposit guttae and they become guttae? I think, my personal thoughts is that I think that there's an abnormal secretion of extracellular matrix from corneal endothelial cells that ultimately leads to guttae formation. And then we know that guttae formation actually, it's a vicious cycle, that guttae can actually cause apoptosis of corneal endothelial cells. So it kind of feeds back into each other. So I think my personal thoughts is that I think it's largely contributed by abnormal extracellular matrix. And then when the corneal endothelial cells die off, that they also contribute to the guttae formation. But there's still some controversy about exactly how guttae are formed.
So can I ask a few questions?
Go ahead.
Really nice talk, Stefan and Marco. Enjoyed it very much. So I have some molecular questions. So TCF4 has an expansion in it.
Yes.
Obviously, it's a transcription factor. So I'm wondering how that expansion affects the binding of TCF4 to downstream target sites. So has anyone done ChIP-seq or cut and run and looked at whether the binding sites of this transcription factor change in Fuchs when you have this expansion?
No, so I don't think anyone's actually looked at binding sites. So, so there's actually some controversial, I mean, there's some contradictory data looking at maybe just overall TCF4 expression. Some studies have showed that there's actually no difference in TCF4 expression, and some have shown that there's upregulation, some downregulation of mRNA expression. Interestingly, this repeat expansion is in an intronic sequence of the TCF4 gene. But specifically those experiments looking at binding sites downstream from TCF4, I don't think it's been done for Fuchs.
Okay, and then the other question I had is about this. It looks like maybe you have cellular heterogeneity of these endothelial cells. The ones in the periphery might be different.
That is correct, yeah.
Center. So have you considered doing spatial transcriptomics? So to look at actually almost at the single cell level at the transcriptome of cells that are peripheral versus central? I think that would be nice.
Yeah, absolutely. And that's something that, I mean, part of, I think we talked about it already. Part of one of the interesting questions that I want to ask is, and maybe there's a different way of approaching it, is doing single cell RNA sequencing, looking at maybe just normal corneal endothelial consciousness at the Fuchs. And I think looking at, but you know, definitely one of the challenges is that you really need to isolate and cells at a single cell level to do that analysis, and that could be a little bit challenging with the Fuchs tissue. So this spatial analysis, I remember we were talking about at some point, is I think very interesting and something that I think would be interesting to really pursue it, because I think it hasn't really been completely investigated. We know that that the peripheral environment of the corneal endothelium compared to the central environment of the corneal endothelium is different, and there, you know, and I suspect that there's some sort of, I mean, there's some sort of stem-like corneal endothelial cell in this periphery that's been identified in this quote unquote transition zone. But in terms of actually molecularly characterizing it, I think that still remains to be determined.
Yeah, that could be really nice. And then just my last question is about the chromatin. Have you done ATAC-seq or considered doing ATAC-seq? I think it would be nice to look at whether that changes as well, the chromatin structure.
Yes, I think that's a very good suggestion. I have not done that, and I don't think it has been done in the literature either.
Just a reminder to everyone that for those of you who missed the QR code on the slides, the link to the evaluation is also posted in the chat box. If you just scroll back to Amandeep's comment, there's a link to the Google's document for the comments. So it's shortly after 8:30. Again, thank you very much, Stefan, for bringing us your work. Really, really interesting stuff. And I hope to see you soon at Sunnybrook. Okay.
Thank you.
Next week, we have Dr. Kaplan. Bye-bye, everybody.